Hello, everyone, and welcome to today's GEN webinar. It's entitled Monitoring Aggregation Levels of Biosimilar Monoclonal Antibodies Using Mass Photometry and Size-exclusion Chromatography. Our webinar was made possible through sponsorship from Refeyn. I'll be your host today. I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotechnology News, which has been the leading source of information on new tools and technologies for the life sciences for over 40 years. Aggregation is an important quality attribute of many protein-based biopharmaceuticals, including monoclonal antibodies, that can influence product efficacy and safety. As such, monitoring of aggregation levels is essential. Size-exclusion chromatography is widely considered to be the gold standard analytical tool for assessing nanometer-sized aggregates and is often combined with multi-angle light scattering to enable the determination of molecular weight and size. However, the technique can be complicated by several factors, including column and mobile phase optimization. Mass photometry is a novel, label-free analytical tool based on detecting light scattered by single molecules, enabling the monitoring of the molecular mass of biomolecules in solution. This technique can analyze small sample volumes and concentrations under native conditions in just a few minutes. Our presenter is going to talk a lot more about these technologies shortly. In fact, let's meet our presenter for today's event, Dr. Gareth Rogers. He's Director, Product Management and Manager Advanced Development at Refeyn. The company pioneers analytical instruments that feature mass photometry. Gareth will discuss how both size-exclusion chromatography and mass photometry we use to measure aggregation levels of trastuzumab, a monoclonal antibody, and several trastuzumab biosimilars. The results of this study demonstrate that size-exclusion chromatography and mass photometry are complementary, highlighting the usefulness of mass photometry as an orthogonal technique for monitoring aggregation of biopharmaceuticals such as monoclonals. Before we start our webinar, I want to remind the audience that there will be a live Q&A session that will begin right after the presentation ends. Gareth will be available to answer as many of your questions as he can, so simply type your questions into the Q&A box and hit submit. So if everyone is ready, Gareth, the floor is yours.
Right, hello everybody. My name is Gareth Rogers. I'm a Director of Product Management at Refeyn Limited. So thank you for joining this webinar today. I'm going to be talking about the monitoring of aggregation levels of biosimilar monoclonal antibodies using mass photometry and size-exclusion chromatography. So just to give you a brief overview of what the presentation will entail. So I'll give you an overview of what mass photometry is all about. So we'll touch upon the fundamental principles of the detection technology, and we'll have a look at some applications as well. We'll then have a look in a little bit more detail about antibody aggregation. Why is it important? Why are biopharmaceutical companies and biotech companies that develop therapeutic antibody drug products, why are they so keen to be able to detect and quantify aggregates of their therapeutic antibodies? And then I'll also run through some data that one of our industrial collaboration partners acquired for analyzing trastuzumab biosimilars, utilizing both mass photometry and SEC. And then we'll have a look into some of the considerations for both techniques, because as I'm sure a lot of the people attending this webinar will be more than well aware, there's a very broad range of bioanalytical tools out there for looking at things like this, and often each one will have a series of pros and cons. So it's certainly quite common where the detection of aggregation and other critical quality attributes is concerned is to utilize a suite of different analytical techniques. So what is mass photometry? Essentially, just a very innovative biophysical analysis tool that has a range of different applications. So it's primarily used for looking at the analysis of complexes, for interaction studies, and also for the purposes of sample characterization. So what it enables you to do is you can get a very rapid, accurate mass measurement, and it's a single molecule method that's also label free. So that means you don't have to do a massive amount of sample preparation before you can start having a look and characterizing the samples that you're interested in studying. And you can look at your molecules of interest in solution, and as I said, in their native state. And this is particularly a powerful technique because it allows you to gain access to subpopulations within a sample, and that might be important if you're interested in wanting to determine stoichiometry and also the oligomeric states of a particular sample. So say if your molecule of interest maybe functions as a tetramer or trimer, and you're particularly interested in looking or being able to isolate and study that particular oligomer, you can do so. It's particularly powerful at being able to monitor the formation of larger macromolecular complexes if they have a complex multi-step process of formation. It's also particularly good at detecting low abundance species as well. So because it's a single molecule technique, each individual molecule or particle within solution, even if it's in very low abundance, you can still detect it. So the technique has a very broad dynamic range. And it's particularly powerful for characterizing and monitoring sample heterogeneity. And that's one of the things that we'll be touching upon later in the presentation with regards to antibody aggregates and other size variants. So even though we're going to be focusing on antibodies today and primarily protein-based molecules, the technique of mass spectrometry itself is not just restricted to looking at proteins. You can also use it for working with small viruses, AAVs, particularly commonly studied utilizing mass spectrometry to determine if you have an empty full ratio and being able to optimize things so you can get more full AAVs, which are particularly important if you're wanting to introduce a DNA payload into a patient for treatment. You'll want less of the empty AAV particles and more of the full. You can look at nanostructures, nucleic acids, and also larger macromolecular assemblies as well. So it's not just necessarily tied to the analysis of proteins. So how does it work? So this is just a very kind of top level summary as to what it's all about. So if we start at number 1 here, so hopefully you can see my laser pointer. There's a glass cover slip here, and here we have some molecules in solution. And we have laser illumination in this system. So it's just a 488 nanometer laser, and that will kind of come up through the sample. And what we get, if we move to number 2 panel here, is that there is a glass water interface. So you'll have a large amount of reflected laser illumination, but some of that incident laser light will also be scattered by the molecules in solution. And what happens is this technique relies on the interference of the reflected light and the scattered light. And what you then get are these kind of classic airy disks or point spread functions. And so for some of the more eagle-eyed amongst you, you'll see that some of these contrast patterns, interferometric contrast patterns, have different intensities. And what we know from a whole range of different studies that have been performed over the years is that there is a very good relationship between contrast and mass. So the contrast scales very linearly with the mass of the biomolecule. And if we shift over to panel 4, the output data is essentially a mass histogram. So we can count each of these individual molecules, each of these individual particles. So each of those is detected and counted, and that gives rise to this mass histogram that you can see here. So if we begin to move on to what can we use this for in a little bit more detail. So usually where biophysical characterization is concerned, there are kind of 3 broad areas of interest that people want to be able to use these tools for: to analyze complexes, to look at how different species may interact, whether it's an antibody-antigen interaction, and being able to pull out maybe a dissociation constant, and hence get an idea of the affinity of that interaction. And also sample characterization, whether it be just a very simple purity check, or if you have something that's a drug that you have developed and you just want to check maybe how stable it is in a particular formulation, so maybe part of a formulation screening assessment. So how stable is it within that formulation? Or if you want to monitor purity over time. So over time, where, say, drug product shelf life is concerned, is the species that is the active therapeutic component still there if we store a drug product for a long period? So within that, there are, particularly from the perspective of industry, we can break this down into broad areas of research and discovery, process development, and quality control. And so classic value chain in the biopharma industry, and often mass photometry can be deployed at various points here. So in research and discovery, for trying to get a fundamental understanding of how biomolecules interact, how they behave. Also very valuable during process development. Once you have a product and you're wanting to optimize maybe the product itself, or to optimize the process for the generation, manufacture of the product in question, and also for quality control. So again, as I mentioned earlier, for looking at things like sample purity and quality. So for research and discovery, there are just a series of questions that we're primarily interested in. What are the compositions of the interacting species? Does the expected complex form? What is the stoichiometry? How does it assemble? And how stable is that complex, and how strong are the interactions? For process development, we might be interested in how stable is the formulation, what conditions can be changed, manipulated in a controlled manner to further improve product performance, and what conditions are best for producing the desired protein, as an example. And then for quality control, is the sample in the requisite form or state in order to be able to pass along into the next stage of analysis or development and production? And finally, does the product meet its CQAs, or critical quality attributes? So mass photometry itself, as I've mentioned already, you can use it for looking at the analysis of complexes. So here's just a very simple example, looking at a large complex, in this case, the bacterial ribosome. So fundamental question is here, can we look at whether it's, what impacts upon its formation of the full complex? And what we can see on the right here is that we only get the complete ribosomal complex in the presence of magnesium ions. So in the absence of magnesium, we only have the subunits, whereas when we add magnesium to the buffer, we get the formation of the complete ribosomal complex. And as you can see, in this particular instance, the full complex is 2.1 megadaltons in mass. And this is just a nice little image here that kind of illustrates what I was saying earlier about the contrast and how that kind of scales linearly with mass. So in this case, this kind of lighter contrast pattern, that would probably be one of the ribosomal subunits, and this more intense contrast pattern would more than likely correspond to the larger subunit complex. So it's very intuitive to start seeing things or trying to understand what is within your sample, even just by looking at the images as they're being acquired by the system. You might also be interested in understanding the conditions under which a complex forms. So this is another example, looking at a chaperone complex. And what we find is that in the absence of ATP, only sub-oligomeric species are detected. And upon the addition of ATP, the chaperone then assembles into these larger 14 complexes, so the full chaperone. You can also use it for looking at oligomer formation. So this is a HIV antibody 2G12. And we can see that in solution, even in just a very small amount of sample in a relatively small volume, we can see that it forms a range of different oligomeric states. So the monomer is clearly the predominant form, but we can also see dimers, trimers, and tetramer. So we can clearly identify that the most abundant species is the monomer. But even though there are many more monomeric species in the sample, because of the broad dynamic range, we can still detect those species that have a lower abundance, such as the tetramer. So the monomer is approaching approximately 2,000 counts, and there are only a small number of counts of the tetramer, but nevertheless, we can still detect both. You can also utilize it for not necessarily just protein-related work. In this instance, we're looking at polymers and vehicles for drug delivery. So in this case, we're looking at telodendron micelles, and these were used as a means of being able to effectively solubilize a water-insoluble drug called fulvestrant. And the group in question here were interested in using mass spectrometry to study these micelles in a little bit more detail and to use it as a way of being able to assess how effective or how well the micelle has loaded with the drug of interest. So we have empty micelles shown here in the top couple of panels, and then in the bottom panels here we can see that these micelles with fulvestrant present in different proportions. So we can see that there is progressively more loading of fulvestrant as we increase the or increase the abundance of that particular drug. We can also look at interactions, as I mentioned earlier. So here we have kind of a classic antibody antigen interaction study looking at Herceptin and its antigen, human epidermal growth factor 2 receptor. And if we look at the top panel here, so these are individual separate mass spectrometry measurements of just the antigen alone, and then just of the antibody itself, Herceptin, which is a monoclonal antibody. Herceptin has 2 binding sites for the antigen, and if we then look at the bottom panel, by manipulating the ratios, so the mixed ratios of Herceptin with its antigen, we can see different complexes forming. So in an excess of antigen, we can now see that the 2 binding sites are populated, and we only have a small proportion of antibody bound to a single antigen, and we have very little free antibody. If we have a 1-to-1 mix ratio, we can still see that we have a lot of free antibody, very little complexes that consist of both of the Fab arms containing antigen, and we also see the presence of some species of some antibodies that are bound to just a single antigen. One other thing that you can do following and using this approach is you can do single shot KD. So you can determine a dissociation constant in just a single snapshot measurement, not necessarily having to rely on a titration. And here we're looking at mixtures of IgG and the Fc receptor. So we have, again, as shown on the previous slide, we have the mass spectrometry measurement of the antigen alone and also the antibody. And then if we mix them in a 1-to-1 ratio, we can see that there is a nice complex forming between the antibody and the Fc receptor. And the dissociation constant that we can extract suggests a nice tight binding interaction. If we then repeat this, but we remove the glycosylation of the Fc region of the IgG, so in this case, just removing the N-glycans, what we then notice is that there is a difference now. So there's a 20-fold change in binding affinity. So by removing the glycans from the Fc region, we've actually, this has actually led to a weakening of the binding interaction between the antibody and the Fc receptor, as we can see here. So and from this change, we're able to extract the dissociation constant just by the varying proportions of the different complexes that we see. There's growing focus on bispecific antibodies, and there's a very nice recent paper that came out earlier this year where mass spectrometry was used to characterize high molecular weight byproducts of bispecific antibodies. So this particular antibody, which is a trivalent bispecific 2+1 heterodimeric antibody that serves as a T-cell engager. So it brings a T-cell into proximity with a tumor cell, although it's not been disclosed in the literature as to what antigen epitope this part of the antibody is binding to. In this particular case, there's the classic kind of like knob and hole approach of being able to promote the formation of heterodimers. So just as a way of ensuring that any kind of homodimeric species are not formed, or the formation of them is kept to the smallest amount possible as an impurity. So this particular study characterizing high molecular weight byproducts is very important because it has an impact on product efficacy and safety. And also by doing kind of more detailed in-depth studies here, this can potentially aid antibody design as well, helping to try and prevent the formation of these byproducts in the first place. That's the idea anyway. If we can eliminate their formation at the start, that simplifies the purification efforts massively. So in this particular paper, they used a range of complementary analytical tools. So that kind of ties into the theme of what I mentioned at the start of this presentation, that there is no one perfect analytical tool out there, and it's very commonplace to utilize several different tools that will often bring kind of complementary information, which can be very useful for an in-depth characterization. So in this case, the authors use mass spectrometry, SDS page, Native MS, and a few other mass spec-based approaches. So this is mass photometry data, and this is kind of the purified bispecific antibody. And so we have samples of the antibody monomer shown here, and 2 high molecular weight fractions that were taken from a chromatography column. And just something I wanted to highlight, and I'll come back to this on another slide, is you can see the measurements were performed in 2 different conditions, either in PBS or low pH in the presence of acetic acid. So just by a simple manipulation of the environment, you can extract a little bit of additional information. So what you'll notice is that one of the high molecular weight species, the dimer here, that appears to be quite stable at low pH. So if we add acetic acid, there's very little change in the dimeric species. Whereas if we look at some of the other high molecular weight fractions and also the tetravalent control, we can see differences between PBS versus acetic acid measurements, suggesting that there might be some non-covalent linkages there. So in this particular study, mass spectrometry complemented other analytical tools as part of a multi-method approach to characterize a complex bispecific antibody and the high molecular weight byproducts. So it's a very fast analytical technique, so just under 5 minutes from measurement to analysis per sample, and is tolerant of a wide range of buffer conditions. So in this particular case, the impact of a sample environment pH change can be assessed very quickly without having to do a lot of optimization. So if we move into focusing in more detail on antibodies now. So the antibody-based therapies, think about, we're all aware there's a rapid pace of discovery and development here. And the majority of approved antibody-based therapies are monoclonal antibodies. And I think the first monoclonal mAb was approved by the FDA back in 1986. And I think the FDA had approved their 100th monoclonal antibody back in 2021. So there's obviously a lot of interest, and these drugs have helped a lot of people with a large range of health conditions, where often cancer-related treatments are our main focus, but they've also added clear value to patients afflicted with various other disease types as well. So the majority of the currently approved antibody treatments are monoclonal, but there are also bispecifics, and we'll probably see more bispecific approvals coming through over time now, given the intense interest there. Antibody drug conjugates, the same. There are a few approvals, but that's another very active area of research now. And a couple of the other antibody-based therapies approvals are just antibody-based cocktails. So they're complex biotherapeutics, and from a regulatory approval perspective, there are several different critical quality attributes that need to be factored in. So critical quality attributes, or CQA, they're physical, chemical, or biological properties that must be within an appropriate limit or range to ensure product efficacy, safety, and quality. So I haven't put an exhaustive list here, but some of the more common ones relate to aggregation, glycosylation, deamidation, oxidation. There'll be, certainly the conference where I am currently, which is the American Society for Mass Spectrometry. There's a lot of focus on charge variant analysis as well. So where biosimilars are concerned, from an analytical or characterization perspective, it's important for the manufacturer of the biosimilar to create something that is very comparable to the original reference product with respect to safety and efficacy. And any variations in the critical quality attributes will need to lie within the range that was set for the original reference product. So as I move on to talk about the results of the small-scale study that RIC Biologics completed, they focused on looking at Herceptin, which was the original reference product, and they had several biosimilars that were based on Herceptin as the original innovator drug. So a biosimilarity assessment would involve physicochemical and functional studies. So on the physicochemical side, looking at the primary structure, higher order structure, or trying to identify the presence of impurities such as fragments. And on the functional side, for example, just looking at target binding. So such assessments do require the use of a range of analytical tools, and often orthogonal analytical tools. So looking at the same attributes, but using a technique that has a different principle of operation. They are considered valuable and important to characterizing and building up a comparative profile, a comprehensive comparative profile of CQA data for the biosimilar relative to the innovator drug. So antibody aggregation won't come as much of a surprise to a number of the audience members that it is often considered to be CQA, and that's because it does have an impact on product efficacy and also product potency and product safety as well. So there is a risk that these aggregates pose a safety risk because they are potentially immunogenic. So what is an aggregate? And this is a question that I'm often asked, and it's certainly not just necessarily tied to large particles. Certainly in the antibody aggregation detection space, it could be a dimer or trimer to those larger particles that would consist of many monomer units. Aggregation can be reversible, irreversible, and certainly where antibodies are concerned, antibody-based therapeutics, they are subjected to various stresses at multiple points during their production, but also during shipping and storage of the formulated drug product. So I've put an example here. So typically where purification is concerned, a lot of these will be purified. At least one of the steps will involve a protein A affinity chromatography column, and to elute the antibodies from that column, they would have to, or the elution would take place at a low pH, typically a pH of 3 to 4. So that's quite a low pH, and exposure to that low pH potentially increases the risk of aggregation because it does stress the antibody. Also, formulations, purified antibody. Even after you have a formulated drug products that has a very high proportion of monomer, that purified antibody may also undergo aggregation over time. So these, just ending up with a formulated drug product, that's not just the end of the story. These things will need to be monitored over time as well to ensure that certainly from the perspective of aggregation, there are no aggregates building up over time in that formulation. It can have an impact on whether a drug substance is stored as a liquid formulation for administration to a patient or as a lyophilized powder. So it really is kind of far-reaching. Where there's the introduction or injection of antibody therapeutics is concerned, there are the majority are kind of introduced intravenously, but more antibody therapies are being introduced subcutaneously. That often necessitates formulating the antibody at a very high concentration to introduce it in a smaller volume. So again, formulating at a higher concentration, that comes with the risk of more antibody molecules being present, more likely to be interacting, bumping into each other effectively, which again increases the risk of aggregation. So basically, the main message is that detection and quantification of these aggregates is clearly important at many stages in their production and during the lifetime of the drug product. So if we shift over and we'll have a look at the biosimilar study that was completed by RIC Biologics. Our RIC group have been around for over 35 years, and they provide a range of in-depth analytical services to the biopharma and biotech sectors. We have a lot of expertise where SEC-based approaches are concerned, and also mass spec. So SEC, one of the most frequently used techniques for characterizing antibody aggregates. It is probably considered to be the gold standard analytical tool in that space. It's column-based separation, and there are usually a range of different detector options. So moles, multi-angle light scattering, UV detectors, and also differential refractive index detectors. So SEC-UV data shows absorbance versus elution time, and typically the larger molecules in a sample will elute first. UV absorbance values of the sample will be a combination of its concentration, but also its UV absorbing properties as well. So the molar extinction coefficient, and that's something I'll come back to further on in the presentation. And in comparison to mass spectrometry is not column-based, and it is a single molecule technique, as I described earlier in the talk, and it provides a particle count versus mass. So hence the peak intensity in this case is simply the absolute number of particles. So this is I'll flash this up very briefly because this is the setup that RIC used to look at a series of biosimilar samples and the original reference product Herceptin. So for the SECMALs, they had a multi-component system with parts from Agilent Technologies and also Wyatt, and they used a SEC column from Waters. So you can see the how this particular experimental setup took place. The injection mass, which is something I'll come back to for SEC, was microgram of materials being loaded, and the UV detection with the diode array detector. We're looking at using a detection wavelength of 280 nanometers. So looking at the effectively the absorption of tyrosine and tryptophan amino acids within a protein. So probably as we kind of shift onto the table for looking at the setup for mass spectrometry, probably the first thing that will be apparent to you is there's fewer steps, and that's mainly related to there are fewer considerations for setup, say, versus a sec-based system. So often, really, the only thing that needs to be optimized just relates to sample concentration. So in this case, the samples were at 20 nanomolar, and there was a final dilution step in the measurement consumable, which meant that the actual measurement sample concentration was 10 nanomolar. So RIC group used the 2MP, which is kind of like our flagship mass spectrometry system. It's a very versatile mass spectrometer. You can see the mass range here. So the limited detection is around about 30 kilodaltons, and we can go all the way up to 5 megadaltons. As a concentration range of 100 picomolar to 100 nanomolar. It's a very sensitive approach in that you don't need a lot of material. We've looked at force degraded antibody samples for other collaborators previously. And one particular study I have in mind, we were I think working with 5 picogram of antibody per sample measurement. And you can see the mass precision and mass error here as well. We have a look at the data that was acquired by RIC Biologics. So here are representative SEC chromatograms of the trastuzumab originator, which in this case is Herceptin, and 4 selected Chinese hamster ovary cell clones. So they took the supernatants and they went through a protein A purification step initially. And we can, this is kind of like the zoomed out view. And then if we kind of zoom in, there are a couple of things that should be apparent here. So there are four different biosimilar clones. And as you can see, the main differences relate to the high molecular weight peaks associated with the dimer of trastuzumab. So the dimeric, or the presence of dimers are particularly pronounced in clones 3 and clones 10. And as you can see, very small amount of dimer from the original parent molecule Herceptin. So just performing a sec-mols analysis of the trastuzumab reference product, and just one of the clones, in this case, CHO clone 10, we can see that the reported molecular weight is very comparable between the originator or parent drug versus the biosimilar clone. So the monomer molecular weight is 146.5 kilodaltons for the biosimilar and 146.6 for the original reference product, and the mass of the dimer is very similar as well. If we shift over to looking at the mass photometry data, so looking at the same samples as were analyzed on the sec based system, we're able to pull out the monomeric mass and also the mass of the dimer as well. So in this case, it's 155 kilodaltons for the monomer, which is in agreement with a previous study performed by a group in the University of Oxford, where they used mass photometry to look at Herceptin and look at the interactions of Herceptin with its antigen. The mass of the monomer that they reported was very similar to this. So like for SEC, clones 3 and 10 also identified as having the largest dimer fraction as well. And I have a summary slide that will kind of bring everything and compare it on one slide. So although, because RIC worked very closely with their partners, and in some cases, the information that's shared, or it can't be shared because of the nature of the confidential arrangement that RSE will have with their clients. So this is one possibility of data like this. Is it could potentially point towards perhaps needing to optimize or improve the downstream process, or maybe to set up or make some adjustments to the formulation in an attempt to reduce aggregate levels to that of the reference product. Or perhaps in this case, although as I say, this was not disclosed because of confidentiality, maybe the decision was that clones 3 and clones 10 would be discarded. But yes, there are a whole manner of other things that need to be considered, including the functional assessment of those clones as well. Maybe they were particularly good at binding the targets, and possibly that maybe they persisted with those 2 clones, as I say, but tried to then look at changing the downstream process with a view to try and reduce those aggregate or dimer levels to get it to more closely match the reference product. So if we kind of put that data together in a slide, a single slide, rather than you having to try and remember the slides separately in your mind. Certainly with mass spectrometry, the quantification of aggregation agrees nicely with SEC-UV. So as you can see, with regards to clones 3 and clones 10, those have the largest abundance of dimer, and that is also matched by what RIC group reported for the same clones with mass spectrometry as well. So there is nice agreement there. Just a few things to mention that the data is comparable, but there are certainly mass spectrometry, you can get to the results like this in a shorter space of time with less sample. So in the case of some of the SEC-based measurements, the loading of sample was in microgram order, whereas with the mass spectrometry measurements, we're down in the nanogram range. And as I said, in some cases, there are examples where you can, where we've worked with sub-nanogram sample quantities also. For these mass photometry measurements, just running the measurement itself prior to analysis, you're just looking at a minute or so, and then an additional couple of minutes for analyzing the results in our analysis software, Discover MP, and dependent upon the conditions used for the SEC approach. In this case, it took approximately 30 minutes per measurement. So there are things to consider where both techniques are concerned. So as we've seen, you can utilize both for detecting and quantifying antibody aggregates. Where SEC-UV is concerned, the detector response for monomer and dimer is a combination of concentration, abundance, and UV absorbing properties. So that's something that would need to be factored in if doing a truly quantitative assessment between SEC-UV data and mass spectrometry data. You'd need to correct for the UV absorbing properties, and that could be done by normalizing the data for the molar extinction coefficient. And to ensure consistency with regards to the analysis. So for mass spectrometry, it's quite important in this case if you're looking at several different biosimilars and comparing them to the reference product. When you're performing the analysis, defining the same mass interval range for the monomer and the dimer peaks. In this case, it was the mean plus or minus 2 sigma, so 2 standard deviations, and that was used for all data sets. So it's important that when you're comparing different samples that you set the same interval range and you utilize that for the analysis of all of the samples. Where SEC is concerned, there is a column sample dilution effect as well, which should potentially be borne in mind, as aggregates may dissociate as a consequence of this during the SEC separation process. Where mass spectrometry is concerned, sample optimization really just relates to optimizing the concentration of the sample. That's primarily the main, possibly sole thing that needs to be optimized, and optimizing it to bring it in the concentration range of 100 picomolar to 100 nanomolar. So that's certainly something that's different to SEC, because with SEC-based approaches, you are able to work with and load more concentrated samples. So as I said, at several points during this presentation, there are always pros and cons to each analytical tool, but nevertheless, the information that you derive is complementary and gives you a more broad, comprehensive, in-depth insight. For SEC, there are more factors to consider for optimization. Selecting the correct column, maybe column equilibration steps that also need to be performed before you can actually run the samples of interest. And also, there would more than likely need to be some optimization of the mobile phase as well to improve resolution of the proteins and to minimize absorption to the stationary phase. So, in summary, antibody aggregation is a recognized critical quality attribute and has an impact on product potency, efficacy, and safety. Hence why this is often looked at in the creation of any antibody-based therapeutic, and will be looked at throughout the lifetime or production of that particular drug product. It's also something that would be considered, although I haven't touched upon this, and I think there are a few more webinars coming up on the GEN platform in the near future that will look into this, but developability assessments that aggregation propensity is something that would be looked at into the selection of an ideal antibody candidate early in the drug discovery and development process. Hopefully, what I've shown you is both SEC and mass spectrometry are powerful analytical tools for aggregate detection and quantification. When comparing data sets from both, it is important to remember that UV absorbance data of molecules in a sample is a combination of both their concentration and UV absorbing properties. So to compare the two, you do need to normalize for the molar extinction coefficient for SEC UV data to get a proper quantitative comparison assessment between the data sets for both techniques. When comparing data sets for several biosimilar candidates in this particular study, it is important to have a consistent approach where analysis is concerned. So where the mass spectrometry measurements are concerned, using the same mass interval range for monomer and dimer. Equally, there'll be considerations like this where the analysis of the SEC UV data is concerned with defining the parameters for peak integration, for example. Unsurprisingly, both of the techniques do have pros and cons, but the agreement between the data obtained by SEC and mass spectrometry in this study further confirms the validity and utility of mass spectrometry as an orthogonal analytical tool. So where SEC is concerned, AUC is another analytical tool which is often used as an orthogonal because it is column-free and matrix-free. And equally, mass spectrometry also column and matrix-free, and hence it complements SEC-based approaches very nicely, and could be a suitable orthogonal technique alternative. So finally, just that last slide relating to a summary of mass spectrometry is a biophysical characterization tool. So we can see that it can be used for, well, it's fundamentally a single molecule counting technique, has a wide mass range as well as a high dynamic range, and we look at biomolecules in their native state, label-free. So very little manipulation and to the sample itself is needed before you can start capturing some nice data of the samples that you have. It's applicable to many biological questions. It's quick and easy to use, also cost effective. And there are lots of different things that you can use it for. So I'll just highlight a few which are relevant in this case. So aggregate detection, sample purity, measurement of true native behavior, the minimal sample preparation, and the also looking at the oligomeric state. If you're interested in learning more about mass spectrometry, I'd encourage you to go to our website, which has a very large number of different, well, let's say treasure troves of information. So there's lots of application notes that are available to download. We focus quite heavily on application notes because we work quite closely with all of our customers who are almost always involved in some cool and interesting stuff. There's a very large number of publications that have used mass spectrometry over the years. And if you're interested in kind of learning more, I think we have a regularly updated list of all the latest mass spectrometry related publications that you can have a look through yourself. Equally, on our YouTube channel, we also have various tutorials and webinars that, starting with the fundamentals through to some of the more application-based focus, where some of our users are running through how they've used it in their own research. So thank you very much for your time, and I look forward to answering any questions you may have. Thank you.